Welcome to part B of chapter 17, where we are going to drill down into the mechanics of RNA transcription, looking at an enzyme known as RNA polymerase. So in transcription, we have to start the transcription process from DNA to RNA in the proper place. Because if we just mentioned in the previous slide that reading frame is really important, how do we determine what the first frame is? And so looking at a breakout of the codons, and these are the first letters over here. So if you choose A for your first letter, and then you choose U for your second letter, and then you choose G, so you go U, and then you look for the G on the third letter, we find this really interesting codon right here that's flagged, AUG. AUG encodes for methionine. It's the only codon that encodes for methionine, and it also is the start codon. It is the very first one and gives us a potential reading frame. You might encounter, and we saw this in the virus earlier, you saw those ORFs, those ORFs, open reading frames. That's where somebody has scanned the RNA and found an AUG, which means that it's a potential start for a gene. We may not know what that gene does, or even if it encodes a gene, but we certainly can see the beginning of a gene and a potential reading frame. No other codon in all of the 63 other possibilities do we have a start. So here's a way not only to flag the beginning, but also to put it in the proper reading frame. And here's the fun setup. This is a sequence of mRNA for a gene that I studied as a postdoctoral fellow at Indiana University School of Medicine when I was over at IU. If we scan through this, you might find, and if you scan, oh, there it is, an AUG right there. So now we have the reading frame. So here's the first codon, AGA is the second codon, ACG is the third, GCG is the fourth, UCC, and so forth and so forth. There is our start codon. And you can see in this mRNA that we have that there's a lot of nucleotides upstream that are not going to be encoded in that gene. Part of the processing, if you will, part of the way that mRNA works is that we have to set the proper reading frame for translation. And so we have our AUG right here. You remember when we talked about DNA replication, we had DNA polymerase 1 and DNA polymerase 3. Well, for RNA synthesis, we have RNA polymerase. The interesting thing that sets RNA polymerase aside from DNA polymerases is that, first of all, it's making RNA. That's fine. It still reads 3 to 5, synthesizes 5 to 3, but RNA polymerase has its own helicase activity. Remember that we had to have helicase in order to melt the DNA strands, to pry those strands apart. So RNA polymerase, in the sort of the same vein that DNA polymerase 1 had exonuclease activity, in addition to polymerase activity, RNA polymerase has helicase activity in addition to its polymerase activity. It literally pries the DNA apart, it melts the DNA, synthesizes the RNA, and as the transcript is formed, it will begin to, the enzyme complex can actually remove that mRNA transcript, and it allows the DNA to re-anneal once it has passed through the enzyme. It's using the DNA. One strand is going to be the template strand. We're going to synthesize that RNA. One of the major differences in terms of RNA versus DNA, in addition to being single-stranded, is that RNA does not contain the thiamine base. It uses uracil as opposed to thymine. So ACGU as opposed to ACG and T in DNA. So RNA is single-stranded, and it has uracil base, pyrimidine, instead of thymidine. So in addition to that AUG codon, there are other sequences that help us properly transcribe only genes from our DNA. These sequences are often called promoters. That is to say, they're special nucleotide sequences that promote the association of RNA polymerase with the DNA and facilitating transcription of that DNA. On the other end, down at the downstream end, we have sequences that will promote termination, that will facilitate, I should say, facilitate termination of transcription. So we're not just randomly transcribing DNA any old place. There are very, very specific places where that RNA polymerase is going to bind, transcribe, and release. Put together, the promoter, the coding sequence, and the terminator we call a transcription unit, and this is functionally a gene. So a gene contains a promoter, generally contains a coding sequence of some kind, and most often, not always, but most often, has a terminator sequence at the downstream end. So we have the components for regulated transcription of genes at the appropriate time in the appropriate amount. So let's come back to our syntaxin 4 gene again, only this time we're going to be looking at DNA. Notice that we have T's in here rather than U's. Okay. Well, here's our ATG, which is the DNA equivalent of AUG in the RNA. So there's our start codon right there. That's the beginning of our coding sequence. But look at this sequence right here. TATA. That is known as a TATA box, this 4 nucleotide sequence. By the way, you can scan through here, and I doubt you will find another TATA box in this whole area. I doubt you will. There may be one out there, but doubt it. This is a major, major piece of the promoter that sets us up for transcribing the RNA that we're going to create syntaxin 4 protein, which is really, really important for insulin secretion, by the way. So we have that promoter, and that promoter is where the polymerase will bind and initiate transcription. And then we get down to the end of the terminator. Okay, so we're going to start transcribing, and here's our template. We're reading 3 to 5, synthesizing 5 to 3. We work our way down, elongating. As we elongate, that transcript is removed off of the DNA, and the DNA is allowed to re-anneal with itself. And this thing slides along and creates our primary transcript. When we hit that terminator, the RNA polymerase will stop transcribing. It will release the DNA, and the RNA will be released as well. So now we have the anatomy of an RNA transcript and the pieces that are involved, and let's start putting it together in terms of a temporal set of steps. What we have are sort of 3 major steps, and we're going to see this also in translation as well. We have initiation, we have elongation, and we have termination. And you can probably see where this is going. I mean, we have this promoter here, and we have RNA polymerase. Well, we're in bacteria, we actually add one more little protein called factor sigma. And factor sigma is sort of what we call a transcription factor that recognizes the promoter and helps guide RNA polymerase to start transcribing the DNA. So we have the sigma factor, the RNA polymerase. It starts transcribing, and the sigma factor goes away. So then we have RNA polymerase transcribing along during elongation, and then RNA polymerase encounters the stop sequences down in the terminator region of the RNA, and so it separates. So we have sigma factor, we have RNA polymerase, and we have the transcript here binding at the promoter, melting the DNA, and starting elongation. So we're transcribing during elongation, and notice that sigma factor is not bound anymore. Sigma factor is separate and has moved away. When we hit that stop sequence, RNA polymerase dissociates from the DNA, the transcript dissociates from the polymerase, and we have concluded. Semester, we don't have time to get into differential expression of genes, like why you only express muscle genes in a muscle fiber and not in a skin cell or that sort of a thing. But it does come down to the promoter sequences as well as other sequences, usually within a few dozen nucleotide pairs, either upstream or around the start point. But that TATA box plus a few nucleotide pairs, that's usually where we have that start point. In eukaryotic cells, unlike bacteria, in bacteria we have that sigma factor. In eukaryotic cells, we have literally lots and lots of different proteins that can either help RNA polymerase or hinder it. We call those DNA binding proteins transcription factors. Let me give you an example of a really nice example of a transcription factor. The estrogen receptor. You remember when we talked about cell signaling, we talked about intracellular receptors? Well, a lot of intracellular receptors, when they're activated, move to the nucleus like estrogen receptor and bind the DNA and help make it easier for RNA polymerase to initiate transcription. Other transcription factors are, in fact, repressors. That is to say, they make it harder for RNA polymerase to do it. They lower the ability of RNA polymerase to do it. It's not all or none. There are intermediate steps, and it is a wide open field in terms of levels of plus and levels of minus. It's almost like a biological abacus, if you will, that these transcription factors collaborate and then sometimes hinder each other in order to get just the right amount of expression for a given tissue or a cell. So these transcription factors bind the DNA and assist RNA polymerase. Happens to be RNA polymerase 2, but we're not going to talk about any others, so that's the main RNA polymerase here. And when we bind that promoter, we call that a transcription initiation complex. TATA box is crucial in this. It's sort of the road, it's sort of the mile, the marker, but the other signals around in the DNA are the ones that the transcription factors recognize and will bind specifically to in order to work with the TATA box to initiate transcription. A lot going on there. We have transcription factors, we have the DNA, and we have RNA polymerase. Those together form this initiation complex and get us started on transcription. So here we have that TATA box, and we have some other sequences that are not transcribed usually that comprise the promoter. So we have proteins, that transcription factors that will bind various areas of the promoter and guide that RNA polymerase 2 to the point where it will begin initiating transcription. And so you can have a few proteins, and you can see there are quite a few here now. Their transcription factors do a lot of things either to help or inhibit RNA polymerase from binding and melting the DNA to start transcription. But again, the key things are we have a promoter, we have transcription factors, we have RNA polymerase 2. When they all bind together, we have the transcription initiation complex. And those transcription factors and promoters and some other sequences we call enhancers kind of act like runway lights on a runway. If this is the DNA and this is RNA polymerase 2, they're helping either guide in or say, hey, don't land here, land somewhere else. That's how the promotings in other sequences we call enhancers affect whether or not we transcribe a DNA sequence. So some things about elongation that you should understand. RNA polymerase has helicase activity, and so it's sliding along roughly 10 to 20 bases at a time, opening up the double helix and transcribing somewhere around 40 nucleotides per second. You might remember that DNA polymerase was closer to 50 and prokaryotic DNA polymerase is closer to 500. This is a little slower. There's a little more proofreading going on, but plus that helicase activity gives us an average rate of around 40 nucleotides per second. Don't have just one RNA polymerase here. We might actually have several working a DNA sequence simultaneously. And we're, again, synthesizing 5 to 3. So we're adding nucleotides. And so the 5' end of that transcript is coming out, and the 3' is where we're adding additional nucleotides. So here it is. So we have our RNA polymerase. It's reading 3' to 5'. It is adding nucleotides to that 3' end, synthesizing 5 to 3. The newly transcribed RNA is coming out here, and the DNA is allowed to reanneal with itself as the RNA polymerase moves past it. So with termination, what we have is signal recognition, and a little different, slightly different mechanisms between prokaryotes and eukaryotes. So what we have in prokaryotes is that the RNA polymerase will hit this sequence at what we call a terminator sequence. It's like a TATA box, only it's for stopping. And so the mRNA will disassociate from the polymerase, and that's the end of that. In the case of eukaryotes, that RNA polymerase will transcribe what is known as a signal sequence, a polyadenylation signal sequence, okay? It's actually a signal, and I'll explain what I mean in a second. So the RNA polymerase recognizes that poly-A sequence, that polyadenylation sequence, and will transcribe another 10 or so, 10 to 35 nucleotides and then stop. So it doesn't stop right away like in the bacteria. It moves a little bit past it and then stops. Eukaryotic cells, we are going to process this mRNA. And that includes adding a cap at the 5' end. Okay, it's a G nucleotide that's methylated and some phosphates. It's a 5' cap. Remember that polyadenylation sequence? Well, that polyadenylation sequence is not encoded in the DNA. There's a sequence there, and what happens is the RNA polymerase will go about 10 to 35, and then another enzyme will add poly-A's, multiple A's, anywhere from 150 to 250 A's on the 3' end. And these cap and tail prevent the nibblases from degrading the mRNA. So both ends are usually modified. If it's an exceptionally short-lived mRNA, then they won't modify it, but most transcripts need modification in order to survive long enough to have proteins synthesized from them. Don't talk about introns in this particular picture, but we're also splicing out the non-coding sequences, the introns from inside the coding sequence. Here's a quick picture of how the poly-A thing works. We transcribe through the poly-A signal in a few nucleotides after that, and then we stop, so 10 to 35. Well, another enzyme comes along and can recognize that poly-A signal, clip off some of those extra nucleotides, and then add A's without an RNA transcript, anywhere from 150 to 250. Again, I'm not too worried about all the details here, but I thought you should understand kind of how it works. It's pretty cool. So there's that poly-A tail, there's that 5' cap. Now this is the pre-mRNA. It has neither the cap nor the tail, and it has these what are called introns that, again, enzymes will recognize, splice out, and create our final mRNA. So here's a picture of exons. Those are the ones that are the coding sequences. Introns are the non-coding sequences. And you might wonder, why do we have introns? Well, that's a great question. Some of the reason why is that inside these introns we have other sequences that modify whether or not that gene's expressed. In other cases, we will sometimes leave introns in order to create a different protein from the same gene. For instance, your immune system, it actually modifies the RNA transcript in order to make different kinds of antibodies. And so these introns are really, really wild because they do things that aren't immediately obvious to in terms of a mechanistic thing. They may contain sequences that affect whether or not it's transcribed, or they may be coding sequences that may or may not be included in the final mRNA transcript. So we get these tails, we get the cap, and we splice out the introns. So we've added that poly A there enzymatically. We've gotten rid of the introns, and now we have our final mRNA, considerably shorter than the original. Once it's finalized, it's moved out of the nucleus for translation, depending on whether or not it's going to be in the membrane or it's going to be in the cytoplasm or somewhere else. Here we can see DNA to RNA to protein in one picture, and this is occurring in bacteria. So we have our DNA strand here that was shown in blue. They have the caricature down below. So here's our DNA strand. On the DNA strand, we have the RNA polymerases, and they're going along. They're cruising along. Actually, they're moving left to right. You know how you know? It's because the RNA transcript is longer over here than it is over here. You can see they're getting progressively longer the further to the right we go. And on the RNA, there are ribosomes making protein. Now, the proteins aren't sticking out particularly long here. They're being folded. That's why they're kind of clumpy looking in here, because those proteins are getting folded in real time. But you can see, so we have DNA, we have RNA, and we have protein coming out of those RNAs. We call these structures polyribosomes, and you can actually use them as sort of molecular timers in certain situations. We did some experiments in insulin signaling along those things. Really cool stuff. But this is prokaryotic, because you wouldn't see this in eukaryotes. Why? Because we'd make the mRNA and then ship it out of the nucleus. It wouldn't be anywhere near the DNA when it was being translated. So that's how it goes. We have how we create the RNA. We have the anatomy of a gene, including the promoter and the encoding sequence and the terminator. RNA polymerase in eukaryotes, it's polymerase II. We have initiation, elongation, and termination. Again, each thing is simple unto itself, but it does add up over time. So please take your time with it. Get to know the terms. The terms will help you understand and will help make it easier for you to answer questions. So with that, we're going to move into the function of the ribosome in part 3 and or part 4 of this chapter. So thank you for your time. We will pick up with ribosomal function in the next segment of this lecture. Talk to you soon. Bye.